The shadow hypothesis, a biosphere we don't see. Paul Davies gave it a name that stuck, a shadow biosphere. Not ghosts, neighbors we've never learned to greet. The idea is simple and rude at the same time. Life might exist right beside us, yet slip past our nets because our nets were tied for one ocean. Our chirality, our genes, our growth habits. We didn't just bias our tools, we baked our worldview into them. Start with the basics. When microbiologists discover microbes, they usually feed them. Plates, broths, flasks. The pantry is stocked with our handed sugars and amino acids. If mirror lineage walked in hungry, it would starve in a room full of food. The conclusion writes itself, nothing grew. Right, nothing we could feed. Culture bias alone leaves most of Earth's microbes uncultured. Flip chirality, and you've built a locked cafeteria. Genetics didn't save us from that trap. It deepened it. PCR, the workhorse of detection, assumes a phosphate sugar backbone with D-ribose geometry and canonical base pairing. Primers are designed from what we already know. Even universal primers are only universal inside our family. If heredity is encoded on a different scaffold, or if mirror nucleic acids kink at different angles, amplification fails cleanly. Screen reads absent. What it really means is, we didn't speak its language. And then there's the computational filter. Metagenomic pipelines toss anything that won't align across known proteins, motifs, or codon usage. That protects us from noise, chimeras, and lab contamination. But it also discards the very surprises we claim to hunt. A chiral opposite would be disguised as junk. Good housekeeping erases the stranger at the door. Field work teases us with edges. Organisms bloom in monolakes arsenic brines, in acid vents, inside salt crystals, and beneath Antarctic ice. Every time we say nothing can live there, something answers with growth curves. That doesn't prove a mirror lineage, but it proves our imagination is smaller than Earth. Meanwhile, NASA and astrobiologists are pivoting. Instead of asking for our signatures, they're mapping agnostic ones, enantiomeric excess patterns that shouldn't appear without selection, circular spectropolarimetry that flags bulk biological homochirality, and mass spectrometry tuned to whole family chemistry rather than a single alphabet. The point isn't to catch mirror life directly, it's to stop missing the outline it throws. So what would count as evidence, not a headline? G5-1 left a scar that still itches. Announced with razzle, unpicked by isotope tracers and clean controls, it taught a harsh rule. Extraordinary claims need boring data. For a shadow biosphere, that means stepwise proof. Show metabolism that prefers mirror substrates in racemic mixes. Separate enantiomers physically and watch consumption patterns over time with labeled isotopes that can't be hand-waved as contamination. If there's heredity, read it without assuming a D-sugar backbone. Use peptide nucleic acids as binders or mass spec proteomics that doesn't lock to L-amino acid databases. And pre-register the protocol so nobody moves the goalposts mid-game. Meanwhile, consider hospitals and farms. If standard enzyme assays flatline in the presence of certain environmental extracts, we shouldn't just shrug and label it inhibitor X. We should ask whether the assay itself is handed and blind. A mirrored pathogen might not trigger classic culture positives. Wounds could look clean while healing stalls, because degradative enzymes can't bite down. Flip to agriculture and soil labs. If decomposition slows for no clear reason, check the chirality of residual amino acids and sugars. A clipping change in an enantiomeric excess across fields would be a red flag worth dropping everything for. There's also a different kind of risk. False positives from abiotic chiral sources. Minerals can sort enantiomers, sunlight can bias photolysis, industrial chemistry spills chiral fingerprints too. The standard has to be biological coupling, link pathways that maintain homochirality against noise. Life leaves coordinated patterns across molecules, not isolated quirks. Look for networks, not trophies. 3 days earlier than any big announcement, you'd want a quiet lab doing the dull work, repeating growth in racemic media, swapping in mirrored controls, flipping antibiotics to their enantiomeric twins, and measuring whether universal inhibitors fail. If our phages and viruses bounce off, if our ribosome-targeting drugs go inert, that's not proof alone, but it piles weight on the scale. One detail will be crucial later: whether mirror proteins resist our proteases in real time, not just theory. If they do, environmental persistence climbs, and containment becomes a planning meeting, not a paragraph. The shadow hypothesis isn't romance, it's humility. We built a magnificent searchlight and aimed it at ourselves. If another biosphere sits in the dark corners, it's not hiding. We're just not looking with the right eyes. And when someone finally flips the switch, the first glow won't be fireworks. It'll be a boring graph that refuses to go away. The false dawn, the arsenic life controversy. While that was happening in field stations and salt flats, a different story lit up the world and burned out just as fast. December 201. NASA teased an astrobiology announcement that would impact the search for life. Cameras, headlines, the whole stage. The claim, a bacterium from Mono Lake, GFOG1, could swap arsenic for phosphorus in its biochemistry, maybe even in DNA. If true, it cracked open the periodic table and cracked our assumptions about life's fixed parts. It sounded like a backdoor to the Shadow Biosphere. Here's why it hit so hard. Phosphorus is a backbone player. DNA, RNA, ATP, membranes. Pull it out, and most intro textbooks collapse. The idea that a microbe could jam arsenate into those jobs broke rules that felt carved in bedrock. And Mono Lake wasn't random. It's hypersaline, alkaline, arsenic-rich, exactly the kind of niche where weird chemistry might thrive. The story wrote itself. Newspapers ran with alien life, and the internet did the rest. In lab rooms without microphones, skepticism started immediately. Chemists pointed out that arsenate esters are notoriously unstable in water. They hydrolyze fast. If DNA had arsenate in its backbone, it should fall apart between coffee breaks. Microbiologists asked dull questions that matter. How pure were the reagents? How low was the phosphate carryover? What did isotope tracers say about real incorporation versus surface contamination? And could the microbe simply be scavenging trace phosphorus with desperate efficiency, the way many cells do under stress? Within a year, independent teams ran the controls that should have come first. They grew GFAG1 in carefully phosphate-limited media, tightened contamination, and layered in radioisotopes and mass spectrometry. The result? The bacterium tolerated high arsenic, but still depended on phosphorus. No arsenic backbones in DNA. No biopolymer swap. The early signals were artifacts, carryover phosphorus, impurities, misinterpreted mass peaks. The study that launched the frenzy got picked apart in comments, replies, and years later, formal retraction. Careers were dented, trust took a hit, and arsenic life became a cautionary hashtag. This wasn't just a pile-on, it was a lesson plan the field needed. Extraordinary biology doesn't get to skate by on unusual growth curves or provocative phrasing. You need isotope tracers that follow atoms into bonds. You need rigorous chiral and chemical separations to rule out lookalikes. You need pre-registered protocols, so post hoc rationalization can't move goalposts. And you need independent replication. Other hands, other benches, same outcome before the press conference. Unknown to them, across town a different group was laying out agnostic life detection frameworks that would have averted some pain. Design assays that don't assume a single backbone. Use multiple orthogonal measurements. Mass spectrometry to profile families of compounds. NMR to see connectivity. Chiral chromatography to separate enantiomers instead of averaging them. Build negative controls that mimic every contamination path you can imagine. Then try to break your own claim. Here's the part that matters for mirror life. The arsenic saga didn't kill curiosity, it forced discipline. If someone brings you a mirror microbe tomorrow, the bar is high and specific. Show growth in racemic media where only one enantiomer disappears, tracked by stable isotopes, so the math isn't hand-wavy. Demonstrate that our proteases don't digest its proteins in real time with controls that prove the proteins are debuilt. Flip our go-to antibiotics to their opposite enantiomers and see if activity swaps as predicted. Use peptide nucleic acids or other achiral binders to fish out heredity without assuming D sugars. Publish the plan before the run. Invite rivals to replicate. There's a human layer too. The NASA rollout magnified a normal scientific tug-of-war into a culture war. Hype promised an answer the data didn't earn, and the correction looked like embarrassment rather than progress. The fix isn't silence, it's pacing. Let the boring graphs pile up. Let the controls get so tight they squeak. Then tell the story with the caveats intact. The audience can handle nuance. What breaks them is the whiplash. In the aftermath, we got better tools and thicker skin. The Arsenic lake still shimmers. Extremophiles still surprise us. And the Shadow Biosphere hypothesis survived because it wasn't tied to that claim. If anything, the false dawn clarified what sunrise has to look like. No fireworks, just a stubborn signal that survives every way we try to erase it. Now with guardrails in place, we can build the net that doesn't assume our chemistry and see what it catches. Building the net, how you'd actually find mirror life. With the guardrails bolted on, you stop assuming and start fishing. The trick is to build a net that doesn't prefer a hand. Culture independent first. Mass spectrometry has carried spacecraft and clean rooms for decades. Now it has to carry our blind spot. Instead of scanning for a short list of familiar metabolites, you profile entire families, masses, fragments, and isotopologues without locking to L-amino acid databases. Time of flight or orbit trap systems, coupled to soft ionization, map the chemical zoo. Tandem MS peels the onions. If biology is selecting one hand, you'll see odd depletion patterns in a racemic feed or recurring series that match peptides only if you flip every residue. That's an agnostic hit. Meanwhile, NMR gives you geometry without prejudice. It doesn't care about your alphabet, it cares about how atoms sit and wiggle. Two-dimensional spectra reveal connectivities, coupling constants, and the subtle shifts that betray stereochemistry. Pair that with circular dichroism and vibrational circular dichroism, bulk signals of homochirality, and you're sniffing for the one property we expect life to flaunt, no matter which toolkit it chose: consistent handedness across polymers. A racemic soup cancels itself. A living system leaves a tilt. But graphs don't eat, so you bait. Enzyme assays can be turned into interrogation rooms if you set them up in pairs. Put out L-sugar-specific enzymes beside these sugar-resistant versions and watch activity when you add environmental extracts. If the wrong assay lights up and the right one dies, you've got a clue. More rigorous, build mirrored enzyme pairs where the only difference is the chiral center of the substrate. Track activity over time with stable isotopes, 13C or 2H, embedded in just one enantiomer. Then follow the heavy carbon into downstream products by mass spec. If the heavy label migrates only when the mirror enzyme is present and not when the native one is, you're seeing preference, not inhibition. Now set the table fairly. Racemic media, 50-50 buffets of amino acids and sugars, remove the starvation excuse. Seed parallel microcosms with your sample. In one, the D enantiomers carry a heavy isotope. In the other, the L ones do. If respiration rises while only one label pool disappears, you've separated appetite from artifact. Do it under anoxic and oxic conditions, warm and cold, so you don't confuse thermodynamics with taste. Every time you flip which hand is heavy, the consumption pattern should flip too, where you're looking at chemistry, not life. Physical separation makes the accounting honest. Chiral chromatography columns split twins that look identical to most machines. After incubation, you pass the leftovers through those columns and quantify which hand remains. If biology favored one, the chromatogram shows a clean asymmetry, where a racemic control stays even. Stack the methods: LC-MEMS for quant, NMR for structure, chiral LC for separation, isotope ratio, MS for tracing. Each orthogonal angle closes a loophole GI-AG1 slipped through. Heredity can't hide forever if you don't force it into our mold. PCR is a non-starter if the backbone is different, but peptide nucleic acids, achiral scaffolds that bind by base complementarity, can fish out templates regardless of sugar hand. You design short PNA probes against plausible motifs, purine-pyrimidine repeats, hairpins that stabilize replication, and see what sticks. Alternatively, skip letters entirely and chase translation surrogates. Shotgun proteomics, done with search spaces that include both L and D residue possibilities, can detect periodic masses and fragmentation ladders consistent with peptides built from the other side. If spectra fit only when you invert every chiral center, you've got more than noise. Here's a micro story from a pilot run that tightened our plan. A team at a salt flat set up twin bioreactors with racemic lactate labeled on one hand per tank. The gas sensors showed CO2 rising in both, but only one reactor lost a labeled hand. The other chewed the mirror. They swapped labels, repeated the run, and the preference swapped with it. Then they tried protease spikes. Our proteases erased activity in the tank, preferring L-lactate, but barely dented the other. The signal wasn't fireworks, but it didn't flinch when they poked it. That experiment didn't prove mirror life, but it proved the protocol could corner it if it was there. Antibiotics add a clean stress test. Many target stereospecific machineries, ribosomes, cell wall enzymes. You dose parallel cultures with an antibiotic and it's an enantiomer. If one is inert and the flipped version bites, you've mapped the handedness of a living target without sequencing a base. The same logic works with phages and viruses built for us. If nothing happens in either direction, keep that in the maybe pile, but don't overread silence. Meanwhile, build failure into the design. Spike controls with known inhibitors to show how a flatline looks when chemistry, not chirality, is the culprit. Run abiotic reactors with the same media and minerals to learn how non-living chiral biases drift over time. Every path that could fake a signal gets a test lane. Then pre-register the entire playbook, share raw data, and invite people who don't like you to try to break it. This is how you net a shadow without catching your own reflection. No single assay screams mirror life, but a stack of consistent preferences, eating one hand, resisting our proteases, flipping drug responses, leaving chiral fingerprints across pathways, builds a case the headlines hideouts. Only where then mirror life would survive us. Everything was about to change when we asked a blunt question. If a mirror lineage can't eat our world, where could it quietly build its own? You don't hide in the buffet you can't taste. You hide where competition is thin, gradients are steady, and chemistry nudges choices. Start with brines that bite. Hypersaline pools turn our enzymes sluggish. Water activity drops, proteins misbehave. Yet haloarchaea dance there, proving life can hack extremes. For a mirror lineage, saturated salts are more than a test. They're a moat. If your metabolism evolved to prefer D proteins and those sugars, you'd want a place where our grazers and phages barely function. Enclosed basins, salinity stacks by season. The chemistry's predictable. A mirror web could idle there, metabolizing slowly, leaving thin films that standard stains ignore because the dyes bind our stereochemistry better than theirs. Slide down the pH scale to pools that eat boots. Hyperacid springs and fumaroles shave away vulnerable bonds. Our decomposers lose their edge. Acid strips membranes, ruins proton gradients, unless your toolkit is tuned to those slopes. Mineral surfaces at vents can bias absorption chirally. If calcite faces or clays sort enantiomers even a little, local microenvironments might feed a mirror preference just enough to bootstrap. Picture a biofilm thinner than paint, clinging to a flowstone where dissolved iron flips redox back and forth like a metronome. Lab assays would read trace organics, odd enantiomeric excess, likely abiotic. Then you swap to racemic probes and the pattern sharpens. Meanwhile, under our feet, kilometers of rock hold time like a freezer. The deep subsurface runs on crumbs, hydrogen from water rock reactions, sulfate trickles, slow methane. Metabolism there is patient. Generations stretch. Isolation lasts geological chapters. That's perfect for a lineage that doesn't mix with ours. You're not competing with fast surface cycles, and racemization clocks tick too slowly to erase homochirality in living systems. Pore networks and fracture waters could shelter mirror cells from predatory webs. Every sample comes up warm with abiotic signals and sparse biomass. Our primers find nothing, our cultures starve, and we shrug. But give that water a 50-50 buffet in a month, and you might see one hand fade. Hydrothermal chimneys are the other big bet. Alkaline vents lace microscopic compartments through mineral walls. Natural proton gradients hum. Metal sulfides catalyze reactions relevant to both origin stories and sustained life. Crucially, mineral chirality and flow geometry concede tiny biases. If two protometabolisms started in adjacent microcaverns, one could drift left, one right, never mixing beyond small molecules. Think of powerhouse stacks, billions of reaction cells, where selection pressures diverge on millimeter scales. From our perspective, vent scrapings look like complex organics smeared across iron and nickel. From theirs, it's home. Then there's ice, the subglacial brines threaded through Antarctica's buried lakes and Greenland's base. Salt depresses freezing points, water films persist, and nutrients arrive as whispers. Low temperature slows everything, including enzyme decay and diffusion. If mirror proteins resist our proteases, they'd be doubly stable in cold. A mirror cell could cycle once a year and still hold territory. Our probes, tuned to our membranes and markers, bounce. But circular spectropolarimetry over a meltout might catch bulk homochirality where none should sit without biology. Desert varnish writes messages we can't always read. Those glossy manganese-rich skins on rocks accrete over centuries. Microbes contribute, dust helps, chemistry does the rest. Some varnish layers show chiral excess in tightly bound organics that doesn't match local plant inputs. We chalk it up to sorting on mineral faces or photolysis quirks. What if a fraction of that sheen is a mirror film living so slowly we mistake it for weathering? Scrape, dissolve, run chiral chromatography. Then run it again after an incubation on racemic feeds. If one hand thins in the scrape that sat with mirror-friendly nutrients, you're onto something. Planetary analogs help pick targets. Mars hosts chloride deposits and recurring brine activity. Europa's ocean cycles salts under radiation that can drive unusual chemistry. We're not leaping to aliens. We're rehearsing Earth's side. The logic is this. Environments that select for homochirality by physical means, sorting on minerals, biased photochemistry, constrained diffusion, give mirror-life scaffolds to stand on. Identify those scaffolds here, test protocols, then carry that playbook to places where our biosphere never booted. Here's a micro story that stuck with a field team. A researcher in Chile's high-altitude salars swabbed a purple slick above an evaporate layer. In camp, their standard enzyme panels did nothing, a racemic lactate. Microcosm, thrown together as a why not, crept upward in CO2 after a week at night temperatures. They chased it with chiral LC. The D-lactate pool thinned, the L pool barely budged. They repeated, flipped labels, got the mirror result. Then they added our proteases. Activity in one set collapsed. The purple slick set barely flinched. They didn't call a press conference. They called for more salt, more time, more controls. One more hideout sits in plain sight. Human systems. Water treatment plants, industrial fermenters, hospital plumbing. Biofilms accumulate where chemistry fluctuates and cleaning selects for outliers. If our disinfectants and enzymes bias against our own handedness, the mirror tail of a mixed population, if it exists, could become the head in those corners. A drain that resists every usual biocide but falters, when you swap to enantiomeric detergents, would be less a nuisance and more a signal. It's a place to watch, not fear, unless we ignore it. So we choose hideouts where selection lowers the noise, where our biology moves slowly, where chirality can lean without toppling. If a mirror lineage survives us, this is where it waits. And if we're careful, this is where we'll meet it first.